This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 537, recorded on March 1st, 2019. I'm Vincent Racaniello, and you are listening to the podcast all about viruses. Joining me today from Madison, New Jersey, Brianne Barker.
Hi, it's great to be here. We did have a little bit of snow this morning, but it's a little gray out and 0 degrees Celsius now. I'm looking forward to much warmer temperatures, hopefully someday soon.
Couple months,
I guess.
We have the Manhattan heat. We have 1 degree.
Oh, well, I'm on spring break next week and we'll be in the city, so I look forward to that Manhattan heat.
Yeah, it's huge, 1 degree.
Yeah.
Also joining us from Western Massachusetts, Ellen Dove.
Good to be here. It's -2 Celsius here.
Oh.
So we have the rural chill and also a little north of you.
I put up our Tufts episode last week.
Yes, I saw that, and I posted a blog post about our visit to the wildlife clinic.
Cool. Yeah, the video came out very well.
Yeah.
It's really good. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. How you doing?
All right.
We've got 52 Fahrenheit, 11 Celsius, cloudy. And I guess I can't complain given your weather reports, but I'm kind of sick of this cloudy stuff. And it was like I thought it was going to be spring a little while ago, and then that went away. And like Sunday, Monday, Tuesday night, they're forecasting lows that are sub-freezing. So I'm done with this.
We're looking to get 10 inches of snow Sunday night.
Really?
As are we.
We are? Yeah. Cool.
Yeah.
I did a podcast with Michael Schmidt. He's in Charleston, South Carolina. Yesterday was 75 degrees Fahrenheit.
Wow.
Fahrenheit. Also joining us from southeastern Michigan, Kathy Spindler.
Hi, everybody. Here it's 31 degrees Fahrenheit, -1 Celsius. Partly sunny, really pretty bright sky, and we don't have any snow in the forecast for through next Wednesday.
Do you have any on the ground?
Oh, yeah, we have remnants.
Mm-hmm.
But, yeah, sidewalks and streets are dry.
Couple of announcements. ASM Clinical Virology Symposium, May 5th through 8th, is in Savannah, Georgia, and you have just a week to submit your abstract, March 8th. European Congress of Virology in Rotterdam. Abstracts are due today, so that's the end of that one. We'll just erase that from the show notes. The Intel International Science and Engineering Fair needs judges for their event to be held in Phoenix, May 14th through 15th. So what's up with that, Kathy?
Well, I checked on it because it's been in here for a while, and they still need judges in all the categories. And in microbiology, they need at least 50 judges total, and they have 14 so far.
Oh.
So this can include senior level graduate students within 1 year of their degree, postdocs, lecturers, faculty, research scientists at, industry, whatever. And evidently it's a really rewarding thing. So check it out. We've got the link in the show notes.
Yeah.
Now, you have to pay your own way, right?
Yeah.
Okay. That could be a limit for grad students, but...
Well, if they live nearby, it should be.
Yeah, that's true.
There are lots of grad students in Phoenix, I bet. Yep.
And Tucson.
How far is Tucson from Phoenix?
2 hours.
2 hours, yeah. All right. And Kathy, you have another announcement for us?
Oh, right. My former student, Lisa Grolinsky, wrote to me about a pilot grant program for it's in conjunction with their I forget what the number of it is, U19 or some kind of grant that they have. And if you want to work in this pilot research program with the collaborative cross mice and infectious diseases, you can apply for one of these pilot grants by April 12th. And there's a link to their immunogenetics page. And then I also put in the Pic Twitter link, which gives you the information about the actual pilot grants themselves and how to do that. So it's a little bit of money, but you could get going on something. Their projects include things like SARS, Ebola, influenza, chikungunya, and West Nile virus, and these really powerful genetic mice for looking at various things.
So we applied for this last year and didn't get it. So it is competitive, just a warning.
Mm-hmm.
We'll try again because it's a cool program.
Mm-hmm.
Yeah.
All right. Henry writes, Dear Dr. Vincent and everyone involved in this great space for the dissemination of science. That's a great intro. Love it. There are no words to describe how happy I was when I heard that our new paper describing the pathogenesis of distinct flavivirus NS1 proteins was featured in your weekly podcast, TWiV 535. I love the title, Miles to Go Before I Leak. For a young scientist like me, it is very important to feel that we are somehow contributing to the virology field. Even though I graduated as a bacteriologist more than 10 years ago in Colombia, where I worked mainly in another amazing topic, the antimicrobial resistance, I fell in love with virus since I started working with hantavirus and West Nile in Colombia. I consider myself a virologist, and I love this NS1 protein. Indeed, such a scary protein with so much potential in the flavivirus field, and why not in all the virus families that secrete proteins that do stuff, as you well pointed out. NS1 is an incredible multifunctional protein that even before being secreted is moving the cell machinery from the inside on behalf of virus replication. It was for many years used as a diagnosis marker for active dengue virus infection in addition to viremia. Actually, NS1 is the first secreted viral protein, at least so far, the only viral protein described to be secreted by flavivirus-infected cells, even before the virus goes out of the cells. It also binds the complement and many other soluble factors causing pathogenesis. Importantly, it causes dysfunction of distinct biological barriers, such as the placenta barrier, the blood-brain barrier, the liver sinusoidal barrier, the pulmonary barrier, and many more barriers that we are still trying to elucidate. I have to say I've listened to this podcast at least 3 times so far, and that I was so excited to hear that all the right questions were asked, the right comments were said, and also very happy that after a couple of very hard years trying to publish this data, as it was also pointed out in TWiV, isn't it amazing that she remembered the 2017 talk? We were able to transmit our knowledge to so many people, which is, from my point of view, the ultimate goal of doing science. Of course, besides trying to find ways to cure diseases, learn about these beautiful creatures, if we are allowed to call them this way, called viruses, and finding ways to come across future antiviral therapies, and why not vaccine development. I must say I'm very proud of every single human being and also non-human components, reagents and cells count too, right, of what these days we call the NS1 team. So collaborative and so powerful, in many ways speaking, mainly human and scientific. Right now, I'm not working at the Dr. Harris lab anymore, at least physically, because I just moved to Mexico to start a new life journey and my new scientific path. My name is Henry Puerta Guardo. I'm a virologist from Colombia. I studied bacteriology, Colombia, microbiology and cellular biology, Venezuela, and experimental pathology, Mexico. And I'm grateful for this and all your podcasts. Abrazos, Henry. I love it. It's great.
That's a nice, enthusiastic letter. I really dig it.
That is awesome.
So what is the show or the movie or the video where at the end they tell you who they are and what they do? That's a common thing, right? There's no one specific.
Yeah, I've seen that as a common motif where somebody talks, they just kind of do a cold open into the discussion of what they're doing, and then at the end they say, "My name is."
I like that a lot.
That's a nice way to do it.
I really like it.
Mm-hmm.
Kathy, you can take the next one, please.
Sure. Ryan writes, Dear TWiV, the measles is no longer a virology issue. And he puts in a link to the Washington Post article. Amid a relentless anti-vaccine movement and measles outbreaks across the United States, a Texas lawmaker has falsely suggested that antibiotics can be used to treat the deadly childhood disease. Texas State Representative Bill Zedler, Republican, an anti-vaxxer who is promoting legislation to allow parents to more easily opt out of vaccinations for their children, said he had measles when he was a child. When I grew up, I had a lot of these illnesses, Zedler recalled, according to the Texas Observer. They wanted me to stay at home, but as far as being sick in bed, it wasn't anything like that. They want to say people are dying of measles, he added. Yeah, in third world countries they're dying of measles. Today, with antibiotics and that kind of stuff, they're not dying in America. Zedler, who represents an area that includes Arlington, situated between Dallas and Fort Worth, could not immediately be reached for comment by the Washington Post. And so then Ryan adds, this stuff is more political than we think, as this Washington Post article is saying. And then there's another link here.
I had a look at this. This is a short bit on how the vaccination efficacy can wane, and some adults might benefit from revaccination.
Mm-hmm. It's not. I don't know why you can say things that are wrong. Oh, hey, that's what everybody does.
Yeah,
antibiotics, right? And kids are not dying in America.
It's the post-truth world.
Measles fatality is 1 to 2 per 1,000 in developed countries, 1 in 300 in undernourished kids. There's no question that kids die in the U.S. of measles, but of course, this person has an agenda who just wants to use the opposite of the truth to push the agenda. It's very sad. I mean, this measles thing is out of control.
And this individual, my guess is, doesn't give a hoot one way or another about vaccination, but is trying to curry the anti-vax vote.
Maybe
it has become political, and that just drives me nuts. As I've said before, they got this anti-vax group in Texas where their website says nothing about the science of vaccination or anything. It's all about personal liberty. Okay.
I'm waiting for someone whose kid dies from not being vaccinated to sue the anti-vaxxers and say, you told me this was okay. I'm suing you. It'll happen.
It was God's will.
Okay.
Have you read the letter that Roald Dahl wrote? His daughter died of measles, and he wrote a letter a few years ago about how he felt and how he wished his daughter had been able to get the vaccine. And it's pretty powerful.
Oh, I have to look at that.
Yeah, it's very good.
We must have talked about it, no? Roald Dahl.
I don't think so.
Letter. Okay,
did you mention kids of anti-vaxxers are getting vaccines?
Yeah, we mentioned that last time. Yeah, we mentioned that last time, yes.
There's hope for us all.
Why does it have to be politicized? It's ridiculous.
Everything is.
Everything. Oh, my gosh.
The world.
Oh, boy. What a week in politics, huh?
Yeah.
All right.
Crazy.
Let's talk about some science.
Science is...
Hey there.
Here on TWiV, we do not politicize science unless a politician says dumb things about it.
Unless they start it.
And then we will say, like this politician is wrong to say people don't die in America of measles. Give me a break. That's just wrong. We have a snippet, which is really cool.
Yes, yes it is.
It's huge. As Amy in my lab says, this is huge. This is an article in Nature Communications, which I believe is an open access. Yes, it says it at the top of the page, open. Numerous cultivated and uncultivated viruses encode ribosomal proteins. And we have two co-first authors, Carolina Mizuno and Charlotte Guillaumard. And this comes from the last author is Mart Krupovich at the Institut Pasteur. I had Mart as a symposium speaker way back when I was ASV president. It was at 2015. And besides the Institut Pasteur, we have the IGDR, Institut de Génétique et Développement de Rennes, Department of Energy Joint Genome Institute, which is in Walnut Creek, California, Universidad Miguel Hernandez, Alicante, Spain, and Ohio State University. So we have talked multiple times here on TWiV about how big viruses are being found more and more to have components of the translational system. When I started teaching years ago, I used to say no virus encodes any part of the translational apparatus, and that's gone. And we did a Tupan virus paper last year, which encodes everything but the ribosome.
Yep.
And now, apparently, some viruses encode parts of the ribosome.
Even that boundary has been breached.
Oh, my gosh. So that's why this paper is important, because it's the first time someone is showing that there are ribosomal protein sequences in viral genomes, both cultivated and uncultivated. And in Paris, we know the difference between those two, right?
Yeah, uncultivated viruses belch at the dinner table.
Right.
They don't go to the opera.
They don't go to the opera.
That's right. They are the opera.
And so this is, I want to just, for a minute, just say the introduction is nice because they say, viruses seem to be sampling different gene pools that we thought they didn't sample, including ribosomal proteins and so forth, ribosomal RNAs like tRNAs. And they say they sample metabolic genes, but he says they don't sample, they don't take the whole thing. They just get bits here and there. Maybe that helps them to replicate. And so they said, do viruses encode ribosomal proteins? Let's take a look with all the sequences we have. Can we find some evidence? So
so this is a computational paper.
And there is actually some biochemistry later, but there is largely computational. So the first thing they did is they said, all right, ribosomal protein encoding genes. There are quite a few ribosomal proteins that make up the large and the small subunits of ribosomes. So they say we searched available genomes of cultivated viruses, the ones that go to the opera. Now, those are the ones you can grow in culture, obviously. Right. And they used 106 ribosomal protein domains to do this sequencing. And they found 5 of them across 16 viral genomes.
Wow.
Mm-hmm. And so the first one they talk about is this Finkel-Biscus-Riley murine sarcoma virus, which I've never heard of. It's a murine sarcoma virus. It's a retrovirus, obviously, but it doesn't appear to be translated.
Right. Antisense, in fact, in terms of the expression.
Yeah, so they say maybe it regulates ribosomal assembly or something like that. So that's one, and then the remainder are in bacterial viruses, infecting proteobacteria, mycobacteria, and a variety.
And these are not huge viruses.
No, they're not huge. Exactly.
So previously we were thinking, okay, there are these gigantic viruses that carry everything, and they've got some bits of the translational machinery because why not? But these are just some little phages, and they've got pieces of ribosomal proteins in them.
Previously on TWiV, I always wanted to say that.
That's good.
They found a homologue of the bacterial S21 protein in a pelagiphage, a myovirus. What's a myovirus?
Like T4.
Muscles.
Got it.
It's a myvirus.
This protein is a conserved component of the 30S bacterial ribosomal subunit. And they think this gene was acquired from its host, looking at its the phylogenetic tree of all the proteins.
T4, by the way, anybody who has any knowledge of bacteriophage probably pictures T4.
Yep.
Mm-hmm.
Right? It's the icosahedral head and a long tail. Looks like a lunar lander.
It is the iconic bacteriophage.
Yeah.
It is.
Iconic, yes. You're looking at my pillow there.
I am.
Yeah, my pillow that Jolene gave us.
Oh, yes.
It's on my couch here.
Looks like an opening scene from War of the Worlds.
Yeah, they did look a little like that. However, there are spherical phages too, with no tails and what do you call the neck?
A stalk?
No, it's a tail. It's a tail and tail fibers, right?
Right.
They're filamentous.
They're filamentous too.
Shaped.
Yeah. Then another one, BL9 identified in a mycobacterium phage, which is a siphovirus. What's a siphovirus? Is that spherical?
No, they're similar. I looked all these up.
I knew you would.
They're similar, but I think it's got a shorter tail. I'll have another look.
How do they get through the mycolic acid?
That short tail, man. That short tail. Is that a barrier?
Well, it is for a lot of things, that's for sure. I mean, mycobacteria have a special coating on their outside.
Siphovirus, in fact, has a longer tail.
It has a sharp spike.
Maybe that's how it gets through.
A sharp spike.
That's the right answer, right?
I guess.
That would make sense.
We might get mail.
It's fine. We do have a few.
Just don't make it hate mail, that's all.
The spike is really amazing. It looks like just like a spike. The ones that have had their structures solved, they're just sharp as anything. They come to a point. Then another ribosomal protein, BL12, in 7 phages infecting proteobacteria. And they believe this sequence has been acquired by different phages on multiple independent occasions. So this is becoming a theme that the phages are getting the ribosomal genes from hosts, and in some cases they get fixed. They remain in the phage genome.
Yeah, I have a question in that sense then, because when a virus acquires something from its host, and it's not useful, like you said, it was sampling from various areas, it doesn't keep those things, right?
Apparently not.
But it did
I think
keep
we're gonna get
these things.
These are conserved.
They are conserved. There's a lot of energy that they spend making them.
The things that came and went, we don't know about because they're not there anymore, right?
We don't. But these are still here.
Yeah, they are.
Hey, Dickson.
Yes, sir.
My buddy Ed Niles says not to worry about ATP. Don't worry about energy. There's plenty of ATP.
Did you say you were worried?
Isn't that what they said about oil?
No, I think what I said was that
It
virus
takes a lot of energy to hang on to these genes, is what you said.
Yes.
But of course,
Don't worry about it.
they're using the host to do it, so it doesn't really matter, right?
I think that a lot of DNA that's not used is kept because it doesn't matter.
Yeah, who cares?
You know?
Yeah, evolution is bureaucratic. It just adds stuff.
Well, then
Actually,
why don't
But
they?
in a virus, there is a cost because if your genome gets bigger, you have to make your capsid bigger, and then things get out of control.
Here. Unless they don't get included in the capsid, they're just part of the
retroviruses in genomes of living things, the endogenous retroviruses, are often mutated because the open reading frames aren't needed, but they remain even though they're mutated beyond translation. So that's a good title, mutated beyond. We'll have to keep that. All right, the last one is the ribosome hibernation promotion factor.
This is the coolest thing. I did not know about ribosome hibernation. I had to look it up.
Yeah.
And what it is 270S ribosomes, total complete ribosomes, complex together to make a 100S complex. And that enables cells to control translation activity without altering the ribosomal pool. So they do this when there are unfavorable growth conditions. So they hibernate.
They just stick them together and shove them off into a corner.
Yeah.
Well, there's an analogy here between viruses and protozoans that insist. Because there's chromatoidal...
I insist.
I know you do, but I'll have to override that right now. Because the cysts of certain amoebae have what's called a chromatoidal bar. And when you look at the ultrastructure of it, it's crystallized ribosomes from the cytoplasm of the trophozoite, which no longer needs those because it's not metabolizing. And so it hibernates them into a very large structure that you can see with a light microscope.
Speaking of which,
but we didn't call them we didn't call them ribosomal hibernations.
It's a good name.
I'll get to this later, but the observation that there were creatures that had arrays that looked like crystalline arrays of ribosomes was one of the things that gave people hope early on that one could actually crystallize ribosomes,
right?
And get a structure.
Absolutely.
Kathy, do you know how they reverse the dimers?
I don't know, but I suspect that, when the conditions are favorable, something must happen to the HPF proteins and the other factors involved in it. And, maybe they're short-lived, and they have to keep getting replenished, and maybe I don't know. I didn't go that far.
I know.
But I felt like I learned I learned my one thing,
that was enough,
when I was reading this the other day, that ribosomes can hibernate.
Indeed.
I bet it's known, but it's an interesting issue. Yeah.
And in the context of this paper, that's an interesting exploitable aspect of them that viruses could use by making similar proteins and tangling up the ribosomes in hibernation when they shouldn't be, and that could slow down host translation. So
I would speculate that the way you get them to unhibernate is to change the environmental conditions, like you do, for instance, when you swallow the cyst of a protozoan that has these. The whole condition changes, the ribosomes become undone, the trophozoites emerge.
I would like to know the molecular mechanism.
The other thing that might happen.
You were 3.
It's as far as I can go with that one. Thank you.
The other thing that might happen from the viral perspective is that the cell may use ribosome hibernation as a defense mechanism.
Right.
Yeah.
Right.
Which could
And
be
so
overridden by the virus. So it could either hibernate or wake up ribosomes depending on the conditions of the infection.
Yeah, as a matter of fact, they did not. These homologies are not like 80 or 90%. We're talking 40, 50% in some cases. And it seems to me there's even potential here for this thing to be a phony hibernation factor.
Yes.
That could be, that can actually interfere with hibernation.
Mm-hmm.
Yes.
Makes sense. That would make sense.
Oh.
But of course, what makes sense to us necessarily is not what viruses do.
Yeah, we're speculating.
And this is another example where the transfer of these HPF genes from the host happened, and they think it's recent, relatively recent, which means probably 1 million years ago, or less than 1 million years ago. All right, so those are the cultivated. Then they have sequences where we don't have viruses, but we have lots of sequences. They searched 425,000 viral contigs or contigs, whatever you prefer, since it's a fake word, for the same in the same.
All words are fake.
Yeah, I guess.
Thank you, Ellen.
Don't tell a blue whale that.
Well, contigs, I always thought of as contiguous, and that's why
I used to say contigs, but that was a long time ago. In the old days, nobody criticized how you pronounce it.
No, you're right. Now you don't get your grant if you don't make it right.
13. Go ahead. What were you going to say, Kathy? I sensed.
Because there were podcasts before podcasts.
Yes.
Or,
Your conversations were limited back then.
Right.
And, you saw mostly the same people over and over again. 13 putative ribosomal protein genes in 1,400 contigs. And they were able to figure out the hosts for 5% of these. And one of the ways is that you could look at CRISPR spacers. You can match hosts and viruses. It's very cool.
Wow.
Yeah.
Very cool.
I think it's interesting that this bacterial S21 shows up prominently in those. That seems to be a common theme or a popular theme.
Yes. It's three of them, including HPF, were in cultivated and uncultivated phages.
Before we get too much farther, I just want to remind people that when we have ribosomal proteins, we talk about them with S and L, depending on whether they're from the small or large subunit, respectively. Mm-hmm. And then they also, in this paper, tell us about putting lowercase letters, B for bacteria, E for eukaryotes and archaea, or U for universal in front of those. So if there are subunit proteins that are common to all of the kingdoms, then they call them universal. So that's what the S and the L mean when we talk about that.
So BS21.
Bacterial S21. Small.
88% of the pool. That was the dominant one. And they think these BS21 encoding viruses are mostly from aquatic samples. Mm-hmm. So they say, maybe that provides some advantage to aquatic bacteriophages. Is that like they have swimming contests?
Yes,
I think so.
Aquatic bacteriophages.
Is there? Maybe they looked at this, but could that be influenced by a bias in available sequences?
Sure, could, I guess. I think you have a lot. You have a lot from water. Yeah. Yep. It's easy to get water.
Yes.
It's less easy to get other things.
I mean, it's easy to get soil too.
Like cockroach feces is hard to get.
I don't think so. Not in my apartment.
Let's just set you up.
You did. I bet.
10, which were found in uncultivated viruses, were not found in cultivated genomes. So they got 10 new ones there. And so so that's good. That's a good summary. They say the emerging picture is that these genes are acquired through random sampling of the host, and in some cases become fixed. Most viruses, over 99%, have only a single ribosomal protein. There's one that had 2. So you can't make a ribosome. But what they say is these viruses are tweaking ribosomal functioning. I like that.
Yeah.
Tweaking it.
I would have liked to see if they don't seem to have any information here about rRNAs and whether there are any ribosomal RNAs that have been picked up. And that would be kind of interesting since they're the catalytic part of the ribosome.
Yeah.
Right.
I bet they're looking, and that's the other paper.
Yeah, that would be very cool. But so with the proteins, it would be the tweaking.
So I was just going to say, they comment that most of the viruses contain only a single ribosomal protein. So they're a long way from getting to a ribosome.
Yep.
Yeah.
And also in this table 1, they talk about ribosomal protein domains, and the coverage identity is not ever, or the coverage is never close to, well, there's some 85%. But so they're also not saying that the whole protein is there, although they go on and do some experiments with whole proteins. But for some of these, they may be just sort of like the equivalent of pseudogenes or something.
Yeah, you're right. Yes, for sure. I think that they're not all whole proteins. That's right. Yeah.
So did they? I'm a little confused here because we have lots of samples that came from that are designated in this figure 2 as coming from humans or other animals and maybe plants, but we're talking about bacterial ribosomal genes. So are these in phage that came from humans or other animals, or are they somehow eukaryotic viruses encoding bacterial proteins? There was some comment on this in the paper, but I forget about it.
I don't think they know, right? Because
they are uncultured. They are uncultivated.
Yeah.
Right.
Could they be from the microbiome?
Yes. Right.
Where there's lots of bacteria and phages?
Exactly. There could be phage in these samples.
Yes, for sure.
Or, I mean, another thing is they are looking at proteobacteria in a lot of these cases, which is the most closely related organisms to mitochondria. So could this be related to the mitochondrial ribosome as well?
I do note that none of these have the lowercase e identified as eukaryotic. There are bacterial and there are universal. Universal could be in a bacterial virus. Okay.
Mm-hmm.
All right. A couple of other things. They looked to see if there was any bias to acquiring these sequences in temperate versus a lytic phage. Lytic, where they lyse the cell, temperate, they come lysogeny, but they couldn't figure it out. So we don't have enough data to do that. So, fine. The other interesting question, are these genes under purifying selection? In other words, are they sticking around or are they on their way out? So if they're on their way out, there are a lot of mutations, non-synonymous mutations that would change the coding sequence, and they're on their way out. And so they find evidence for they're under strong purifying selection, and they're being retained, and they probably have an important function, although you'd have to certainly test.
Purifying selection, meaning there's a selective pressure that makes the virus maintain the pure or functional sequence.
Yeah. So the ratio of non-synonymous to synonymous mutations, if it's less than 1, that means selection for a functional protein. If it's greater than 1, in the process of being phased out, like you and I, Dickson.
Speak for yourself.
Well, I didn't want to just include you.
Well, don't speak for me. Speak for yourself.
I know. People say that to me all the time. Don't speak for me.
Exactly.
Sorry. Yes, it's true. And finally, the biochemistry. Are these proteins incorporated into ribosomes? So they picked 3 ribosomal proteins in the genomes of cultivated phages. So then they took the genes for these and they put them on a plasmid and put them in E. coli. Right?
Mm-hmm.
And then they purify the ribosomes from E. coli. And they ask, are these proteins being incorporated in them? And they actually do some mass spectrometry. So the BS21 and BL2 are incorporated, but not the hibernation factor. Oh, it's too bad.
Yeah.
No hibernation factor. And then the question is, okay, they're there. Do they participate in protein synthesis? So how would you do that? Well, you'd purify the ribosomes and add them to an in vitro translation. Man, nobody does this stuff anymore. It's so sad, right?
I was thinking, I looked up, out of curiosity, I looked up the methods for their sedimentation analysis of the ribosomes, and they're the same methods that I used in the early 70s.
Yeah.
They haven't changed. It's great.
Yeah, some things don't change.
Some things just work.
They don't have to be changed.
Exactly.
So they could only purify the BS21 ribosomes, right? The other one they couldn't because they had to put a tag into them for purification.
Mm-hmm.
And then they added the BS21 ribosomes to an in vitro translation assay, which, as far as I can tell, they're translating a GFP mRNA, and it screwed up protein synthesis by adding these ribosomes. So they said maybe you need other proteins for proper translation. But I was thinking maybe you need an mRNA that with codons that match the host or closer. I mean, this is a ribosome, I know, not a tRNA, but maybe you shouldn't use a GFP, which is a jellyfish mRNA. Maybe you could use some other.
You could use that to improve your memory, though.
Yeah. Maybe they should use beta-galactosidase.
So
yeah, exactly.
Why couldn't they incorporate the viral equivalent of the ribosomal proteins into the E. coli and then eliminate the phage and just see if E. coli could make proteins based on this new.
You want to substitute it for? Yeah, that would be an interesting. That could be easily done. Sure.
I wonder why they didn't do that.
Well, maybe they tried and
it didn't work. E. coli died.
Maybe it's another paper.
Yeah, maybe.
I don't know. But anyway, these proteins are put into ribosomes, although they seem to be, as we would say, verkakta.
Yeah, verkakta.
Right.
Messed up. I have another question. And that is, have they found any viral coding regions for transfer RNAs?
Yeah.
Because there's lots of those, right?
Yeah. All of them, actually. All of them. How many are there, Dickson?
Well, there are 21 amino acids, so I guess there are 21
So the
RNAs.
Tupan virus has all of them.
Really?
The one last year. And everything but the ribosome. And now, that's the thing I wanted to mention. So they say they're sampling the translational apparatus, but Tupan virus is not sampling. It's got the whole smorgasbord on its plate because it has everything but the ribosome, right? So that's not universally true. It may be generally true.
Is that a cassette that they stole? All the transfer RNAs are in one portion?
No, it's not a cassette. No, it's all over.
So it didn't just sample. It actually selected.
It's a smorgasbord.
It selected.
It went up and took everything. You know those people who do that?
I do. That's a mistake because you'll fill up on all the starches first and you'll miss out on all the good crab legs.
So you have to start at the dessert end.
Tell me what you think. Here, I have an opinion about this statement. Our results further blur the borders between viruses and cellular life forms. I don't agree with that statement.
I didn't like it either.
Doesn't, because if you have everything you need, you're no longer a virus, right?
Right.
I do like the opening line of the discussion, though. The evolution of life is a history of virus-host interactions.
Yeah, it is.
Wow, yeah.
Rich, they did talk about one ES30 domain. It's the second paragraph of results. It comes from, or it's related to Finkel-Biscus-Riley murine sarcoma virus.
Right. The deal there was that it's actually transcribed in the antisense orientation.
That was the one. Right. Okay.
And it's also not the whole protein.
Yeah.
So that sounds to me like a situation where it could be, manipulating the cell's version of that. Right. Expression of the cellular version.
All right, that's it. That's pretty important, I think. But I think
it's a pretty big deal.
I think we're, as they say at the end, the more we look, the more we're gonna find. Yep. You should never say never.
They've now crossed the last unbreached boundary between the cellular and viral kingdoms, as they say.
Is that really the last unbreached boundary?
I don't know.
So we have some viral genes involved in metabolism. We've done those papers. In lipid biosynthesis, I guess so.
I think so.
What about the cytoskeleton? Do viruses encode any cytoskeletal proteins?
Good question.
I don't remember, but that could be one unbreached boundary or not, depending on it. Okay. What else goes on in cells?
They just steal the membranes by budding off of them.
Well, yeah, they repurpose them. They make little vesicles inside to replicate their genomes on. I'm wondering if they contribute.
So certainly there's viral stuff for all the replication and transcription machinery.
There are, yes, for the bigger ones, yeah. All right, so we have a paper which is also cool.
Mm-hmm.
It's a letter in Nature. MHC class 2 proteins mediate cross-species entry of bat influenza viruses. And this is a very large collaboration.
Yes.
Between institutes throughout Europe and the U.S. We have the first two authors, Karakus and Thamamongud, which contributed equally, and then we have two, Martin Schwemley and Silke Stertz, who are the two co-PIs. And Silke was on TWiV when I was in Zurich. I was at her institution, and she didn't mention any of this. She's holding it close to her vest until it got reviewed, I guess. Of course, I didn't ask her about it either. Bat influenza viruses. We talked about this a long time ago in an episode entitled Going to Bat for Flu Research, TWiV 173.
Yeah.
Influenza viruses isolated from bats, Guatemala, Central and South American bats. And the cool thing about those, and this is why this paper is interesting, these bat viruses, unlike all the other influenza viruses that we know about, do not bind to sialic acid to enter cells. And so this paper identifies the receptor, and it's important because you want to know, is it a bat-specific protein, or is it something in common to other species, in which case these viruses might be able to infect other non-bat species.
Right. So to bring people up to speed a little bit here who haven't listened to the previous episodes, we talk about flu a lot. Flu is primarily an avian family of viruses, the influenza viruses. They're in birds. And the discovery of them in bats was kind of a big deal because here you've got a mammal species that seems to just harbor flu viruses.
And if you want to learn about flu in birds and seals, listen to the previous episode.
Yes, the previous TWiV is a good one for that.
It was really good. They go out and sample seals, Dickson, and gulls. Do you know how they trap gulls? Do you know what the bait is?
I'd use
dead fish.
Doritos. Ground up Doritos. And then
I had a seagull on the fly grab a whole piece of pizza out of my hand one day.
You're kidding.
It was shocking. And I really wanted that piece of pizza.
Yes.
Wow, that's amazing. Well, the investigator, Nicola, she said they're very smart creatures, gulls. They're very, very smart. She was
survivors. They're definitely survivors.
They use Doritos, and then they put a net on them. Right. And they have a gun that fires the net at long distance.
I've seen them.
I always think of that scene, the seagulls as they're characterized in, what is it, Finding Nemo?
Yes.
Yep.
They all sit around saying, mine.
That's great. That is just great because they do sound like mine. One more story before we get back to the paper. I know Kathy's getting impatient. I saw a seagull on a beach once. There was those red licorice things about 6 inches long. Mm-hmm. Twizzlers, right. So a seagull went up and it picked it up and it swallowed the whole thing. And then it came right back out again. It didn't want it. So funny. And then they fight each other for food that they find, right? Yes. All right, so this paper, what is the receptor if it's not sialic acid? And by the way, the hemagglutinins, which are given numbers in influenza viruses, these are H17 and H18. And so and then, of course, there's a neuraminidase, right? So it's H17N10 and H18N11.
Wow.
So importantly, relative to all of the avian viruses and other viruses that were known previously, those are two new hemagglutinins and neuraminases specific for the bats.
So this is a bat influenza virus or influenza viruses, and they don't use the normal receptor. And as you might expect from not using the normal receptor, they've got different H and N.
Right. Another question of clarification here. Bats and birds, do they have the same body temperatures?
They're not far off, I think. Bats run hot, birds run hot.
Exactly.
They both have increased metabolism.
Yep.
Kathy is still there? No, Kathy's gone.
Oh, no, we lost
I
Kathy.
knew it.
No, I was muted. Sorry.
Sorry.
Oh,
Okay.
And I'm looking up bat body temperature. Oh, and this is in Fahrenheit, 100 to 105 degrees Fahrenheit.
Right.
So,
Right.
oh wait, here's a whole paper: metabolic rates and body temperatures of bats.
Why do you want to know, Dickson?
From 1951.
I'm just curious as to these are two flying entities, and they both share the same ability to transmit viruses.
Yeah,
Flying
they're-
febrile entities, yes.
Yes, there was a paper previously about differences in their DNA damage response that was related to their differences in metabolism.
Yeah, we talked about that.
Of course, bats hibernate, so that might
They do, many.
And what do
And
they
then
do with their ribosomes?
Well, their body temperatures go down, though. That I do know.
I wonder if they smoosh up their ribosomes when they hibernate.
They might.
No idea.
Huh. All right. So they use a couple of approaches here in this paper, which we've talked about before. They're pretty straightforward, but they use a couple of different ones. First thing is transcriptional profiling. So you do RNA sequencing of RNAs, and they use cell lines that are either susceptible to bat influenza virus or not susceptible, meaning they don't have the receptor. It's brilliant, isn't it?
Yep.
Yes.
Remember the mnemonic: CEPT, C-E-P-T, susceptible receptor.
Maybe that's a good mnemonic.
Yeah.
Yeah.
Yeah, I keep pushing that for our students. So
That's good.
Sept receptor. Yeah.
So are we going to talk a little bit about permissivity with this too?
I don't think they use permissivity in this paper. So maybe
They don't, but that's actually based on the type of experiment they do.
Go ahead. Tell us what you're thinking.
So for all of these experiments, they aren't actually using the full H18 and 11 virus. They are using a different virus that has H18 pseudotyped onto it.
Mm-hmm.
So they're not looking at the actual ability of this flu virus to replicate in any of these cells.
That's right. Right. It's just getting in. Right.
Exactly.
Yeah, exactly. I think there's one experiment with a bat cell and a virus, but so transcriptional profiling. So you say what is in the cells that are susceptible and not in the cells that are not. They found 10 genes in all the highly expressed in all the susceptible cells, and 5 encode MHC2 associated proteins, major histocompatibility type 2 associated proteins, alpha and beta chains of HLA-DR and DQ.
Now, MHC2 is the molecule that presents antigens on the surface of immune cells.
Right.
That's right. Largely
Together with a little
APCs,
piece of the protein.
Right.
Yeah,
the
and
antigen.
And a thing that's sort of important is that MHC class 2 is only present on a small subset of your cells, really the professional antigen presenting cells. So it's just your B cells, your dendritic cells, and your macrophages, along with a couple other ones more rarely. So this isn't going to be on every cell type.
So right away, that's weird, right?
It is.
Right.
Very strange.
What's a respiratory virus doing binding MHC2?
Well,
exactly.
Those, I guess, those cells are in the respiratory.
They are in the respiratory tract, but you'd do better off binding sialic acid, you'd think.
But as we'll see, the infection is limited. So
yeah.
All right. So they downregulate these genes by RNA interference, and basically knockdown of CD74, which is a protein needed for processing and transport of MHC2, or the alpha chain, but not the others, reduce entry by H18 pseudotyped viruses or virus-like particles, right?
If there are any people who are very up on their MHC class 2 biology, CD74 is usually known as the invariant chain.
Invariant chain, yes. And then separately they did, so this is, Kathy, this could be reviewer 3. They did a genome-wide CRISPR-Cas9 strain. Well, screen. I don't believe those results. Do another screen. Oh my gosh.
Well, I think that it was 2 different
groups,
people doing, using 2 different.
They had enough
Right.
people to have been doing both of those experiments at the same time.
That's what I figured.
Yeah, I agree. They said, "Oh, why don't we publish this together?" So we'll put.
Yeah,
This
I was even thinking maybe
Go
these
ahead.
started in separate labs as totally separate projects, and then they met at a meeting and said, "Oh, well, let's get together on this."
We may not have to do any more work with
Well,
them at all.
what we did when we were cloning the polio virus receptor, met Eckhard Wimmer at a meeting, and he said, we're doing the same thing. And so he said, well, let's collaborate. So we did. Yeah. Sometimes it works. All right. So they did this genome-wide CRISPR-Cas9 screen.
This technique boggles the mind. Have you guys seen this before? This just boggles the mind.
Yeah.
So they take cells that are susceptible, and they introduce into them a Cas9 gene. So that's the nuclease in the Cas9 thing. Yeah. But it doesn't do anything if it doesn't have an appropriate guide RNA to tell it what to do. And then they transduce those with a library, a lentivirus library of guide RNAs.
Yeah.
Okay, so now you've got a library of cells, all of which have the nuclease, but individually have different guide RNAs, and they're being expressed, since it's a lentivirus, permanently. So what that means is that in that library of cells with a given guide RNA, it's going to knock out a gene. So you have basically a pool of knockouts. Now you infect all of those with a virus that carries this hemagglutinin 18, the H18 gene. It's on a vesicular stomatitis virus, so it's pseudotype. But basically that means that if you have the receptor, you're going to be infected by that virus and die. So the only surviving guys don't have the receptor, and those are the guys you're interested in.
Yep.
How do you figure out what it is? You go to those guys and sequence the guide RNA. That's the magic in the end to me, because that identifies what the knockout was. I can't. I'm stunned. I can't believe it.
Yeah, I mean, it used to take months to knock out a single gene, and now here we're just going to knock out a library of every gene just for an experiment.
Rich, did you notice the name of the data analysis technique they used?
What was it?
Magic, as you said.
So we just wrote a proposal to use this to identify a receptor, and you can buy these genome-wide oligonucleotides. And the cool thing is you go to Addgene, and they have them donated by different people who have fooled with them and said, they do multiple guide RNAs per gene, and they have tweaked it to get it to be effective and no off-target. And they put these libraries there on Addgene, and you can buy them for not a lot of money. You have to package them into lentivirus vectors, of course, but you can go through and see all the different iterations of libraries donated by different people who, figure out in their lab what works. And then I think it's great that they do this. So you don't have to sort it. I mean, say you wanted to make a genome-wide set of 20,000 or more guide RNAs. Man, you'd have to go through every gene and pick them out. It'd take a long time. So these people have done it. And so it's great. Anyway, so doing that, they identify genes that are involved in 4 transcription factors that regulate MHC2 expression, and also the beta chain of HLA-DR. So
you might say they arrived at the same result through an orthogonal approach.
Yes.
Yep.
Yeah, very good. Orthogonal. All right. So next experiments, they make cells where they've knocked out HLA-DRA, confirmed that the protein on the surface is way down. It's resistant to infection with either H18 or H17 pseudotyped virus-like particles, which they used in the first experiment, and VSV pseudotypes as well for both. So both these H17 and H18 bat influenza virus seem to bind to HLA-DRA. And then they say that it prevented infection with authentic H18N11 virus. But this is not a bat cell. This is a human cell, right, that they're using here. So I don't think there's any experiment where they use a bat cell line, if I recall.
Yeah, they do.
Till the end, they do.
Later.
Yep, at the end.
Okay. All right. So they add antibodies to MHC2. This blocks entry of bat influenza virus as well into these cells. And then here's one that I thought Kathy would explain. They confirmed the interaction of H18 and HLA-DR by proximity ligation assay, which we've talked about before. Do You remember?
Oh, I studied up on the other one, the emulsion assay. Sorry.
No, it's okay. Emulsion coupling.
Right.
What's that?
Emulsion coupling assay is shown in one of the extended data figures, and that's where they cross-link their pseudotype VSV with the class 2 antigen, and then they incubate that with antibodies. Well, this is kind of like the PLA thing because the antibodies have DNA,
the DNA
attached to them.
Yeah.
And then, so those can be fluorescently labeled. And then they emulsify for several hours. And then they do drop digital PCR and identify with different fluorescent tags, which droplets got 2 things and which got 1. And then they,
So the PLA, the same thing. You have antibodies against the 2 proteins that you think interact. The antibodies are linked to oligonucleotides. And if the 2 proteins come together, the DNAs come together and they will do rolling circle DNA synthesis. And they'll amplify it and you'll get a signal. It's really cool. It's just amazing.
You have to have an assay. You've got to have an assay.
How can you lie about science?
You can't.
It's just so cool.
Remember, Harold Varmus, when we interviewed him on our TWiV, said you've got to have an assay. And that was his big push, and he's absolutely right.
You have to have an assay.
Got to have an assay.
All right. So then they took human HEC293 T cells, which are not susceptible to these bat influenza viruses. They put in the gene encoding HLA-DR, and this can be infected with their pseudotyped virus-like particles or via pseudotype VSV. But again, as Brianne said, they don't look at actual virus replication, so we don't know.
Right.
All right, what about pigs and chickens? Because, they could be an intermediate. The bats could infect them.
And because that's part of the normal cycle that we think of for new flu pandemics, it spreads from birds to usually pigs and then to humans.
So they got the HLA-DR genes from pigs and chickens and put them in cells and show that they can also, they say H18-mediated infection, but I'm sure it's pseudotyped as well.
Yeah.
Not in, It's H18-mediated. Okay, I guess that's fair. Yeah. And then bats. So they got HLA-DRs from a variety of bats. The big brown bat, the black flying fox.
Little brown bat.
The little brown bat.
Yes, the little brown bat.
This is sounding like one of those dog shows.
And the little yellow-shouldered bat.
And the wire-haired bat.
And the Adirondack bat.
And they get HLA-DR from all of those, right?
Mm-hmm.
So
they point out that those are not
the wire-haired bat. I made that up.
Those are not the bats that are the natural hosts of this virus.
Right.
Yeah.
They don't have sequence information so that they can use the natural hosts.
They have to get the genome, right? Yeah. But the big brown bat and the little brown bat and the black flying fox HLA-DR worked, right, to allow entry of these pseudotypes with the H18. Not chickens, though. It worked for bats and pigs, but not chickens.
Right.
Mm-hmm.
And little yellow-shouldered bat. It's a new sequence. I don't remember what that happened.
Yeah.
Something weird happened.
So that's the one that's the natural host.
Sternia lilium. Okay.
Yeah, and so there they actually looked at infection not in 293 T cells where they put in the receptor, but in an actual cell line from this natural host.
So they put in a transcriptional regulator of HLA, right? Yeah. Mm-hmm. So they overproduced this transcriptional regulator and made both cell lines susceptible. But they couldn't stain for cell surface MHC2. So they think it's working, but they're not sure.
Yeah. So they think they've just increased the amount of MHC class 2 on the surface.
Yeah. So basically, a lot of different bats. Apparently, these viruses can bind to their HLA-DR. Right. What about mice? Mice lie, monkeys exaggerate. I hear that quoted all the time now, Alan.
Oh, good.
It's good. Our mice, so they in cells. It works in mouse cells. In if they produce mouse MHC2, you can get entry of M18. They infected wild-type mice intranasally. So again, the mouse MHC2 is working. So they infect wild-type mice IN. They see good viral replication. And this is a pseudotype, right? Or is it the virus? It's authentic virus. This is authentic virus. Yeah, authentic.
Yeah, and they see, because they're looking at replication. Yeah, so they find viral replication. It's confined to the upper airways in the mouse.
Yeah, and it's confined to the apical ciliary border of the olfactory epithelium. It doesn't go throughout the entire epithelium, right? They don't say what cells it's in and whether it's other than epithelial cells, right?
I'm going to bet they're MHC2 expressing cells.
They must, right? We hope they are. They knock out this C2TA promoter, which I guess would basically knock down MHC2 expression, right?
Yeah, and they also use MHC2 deficient mice.
So they have mice that lack MHC2 on cells that are not derived from the bone marrow, like endothelia, epithelia. Okay, so and then they can't see any virus replication in those mice, so that's good, right? And so they say nasal epithelium seems to be the only site of productive replication, at least in mice. I'd really like to know in bats, though, right?
Yep.
Yeah.
What's going on? What part of the bat? Is it nasal epithelial? Is it respiratory tract? Is it... And where did they get these viruses from originally? Was it from a swab or guano? I don't remember. I'm sure no one will remember. It's too long ago. It's years ago.
Yeah.
Well, maybe we'll get mail.
I have trouble with last week.
I think it's in reference 1 and 2.
So what I don't remember if you talked about is the antibody blocking experiment.
Yeah, but you could repeat it. You could repeat it. It's fine.
Well, I would because I was just thinking with it in respect to the criteria for identifying a receptor. And so that kind of rang a bell with me that, they're kind of checking off the list of things that you need to do. So, okay.
They did most of it. So they showed the genetics, they have the antibody blockade, and they showed the proximity ligation in the other assay, the interaction. Mm-hmm. That's pretty good. Mm-hmm. But what they don't show is the whole replication cycle, which would be nice to have too. Right. So I don't know where these were originally isolated, but let's see if they say in the introduction. 2 novel genomes in bat species. Yeah, 1 and 2. They don't say.
All it says is in Central and South America.
Oh, that answers it. So this is cool because now we know a receptor, and as they say, this still could infect people, right?
Good.
Remind me, is there a human HLA-DR experiment in this paper?
There are some human HLA-DR experiments where they, in figure 2 and in figure 3. So they are looking at
human. There it is. Yeah,
human cells. Yep. Where they take away HLA-DR and they also put HLA-DR into HEC293 T cells.
Oh, this is great. They have the pictures of the animals, so I don't even have to read it. They got a human, a pig, a chicken, and a bat. This is great. And you can see the bar graphs. Wonderful, wonderful. Okay, so it has zoonotic potential.
It does. This paper showed up, I saw on Twitter, right before I went to start teaching about influenza to my emerging infectious disease class. So we ended up talking about it a little bit. And my students were all like, oh my gosh, this is terrible. We're all going to die.
We haven't died yet.
Yeah, I know. And so I sort of had to point out, well, we're using pseudotype virus. We don't actually see this replicating in human cells. We just see it entering calm it down a little.
Could happen, but so far all the influenza viruses are coming from, water birds.
Yeah.
So that's the other question that I have is that if the pig is the mixing vessel, then why aren't there hybrid influenza viruses from bats and birds where both receptors are present on the outside?
It's a good question. We don't see that, yeah. We see just bird.
Do we see natural pig infections with this bat influenza?
I have never heard of it reported.
Me neither.
Only from the 2 original isolations in Central and South America.
So there's the implication that this is an influenza virus that only infects bats? Naturally?
Well, that's a good question. I don't know of studies that have gone to look at the distribution of these viruses in other bat species, right?
Right.
It may have been done. I'm going to do a little thing with H17N10 in PubMed. Let's see what that comes up with. Exactly. And bat. H17N10 and then bat. Let's see how many papers we get. We get 12 papers.
12.
They're making antibodies, blah, 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 infectious viruses. New World bats harbor diverse influenza A virus. That's the original.
I'm looking at that paper here to find out where they got it, and they assayed all sorts of tissues and swabs and stuff, and from the way I read this quickly, looks like they got virus out of a rectal swab and out of intestinal tissue. Okay?
Yeah.
But no other tissues in the virus, in the animals.
So bats migrate, and some do at least, and people harvest those bats and they eat them.
Some
Where do they do that?
In various places, like throughout Indonesia.
Ozzy Osbourne concerts.
All the other, exactly right. Exactly. Batman Returns. I've seen them do that. Bats on a stick. So I wonder whether or not any human outbreaks of influenza can be traced to a migration event of bats where they were harvested and, like bushmeat. This is what that would be, bushmeat.
That's a good question. Yeah. So they have not looked at these viruses in other bats. Those 12 papers look at other things, but that's an interesting question. Mm-hmm. Speaking of bats, I saw a picture the other day of a flying fox with they have
they're huge.
4 foot,
huge.
And the one had its baby hanging underneath it.
Yeah.
While it was flying around.
I've actually seen them.
I've seen them.
Yeah, they're amazing animals.
Yeah. All right. There you go.
They're apparently eaten in the Pacific Rim.
They are.
And
fruit bats are eaten.
What does it taste like? Does anyone know?
Palau,
it's a little like chicken.
Considered a delicacy.
Tastes like chicken.
A little like chicken.
You're right.
It's about the size of New Jersey. That's those.
No one on this podcast has ever eaten bat meat, right?
No,
we don't know that.
Not to my knowledge.
We don't know that. Our listenership.
No. Right, recording right now.
You mean us?
Well, if any listeners have eaten bat, I'd be curious to know what it tastes like.
Yeah.
All right. Let's read a few more emails. Mm-hmm. Rich Condit, can you take the first one?
Okay, I'm looking up
pictures. You working?
No, I'm here. I was looking up flying foxes with babies.
Oh, no.
Okay. Near writes, Dear Twivers.
Indeed.
Greetings from the University of Chicago, where the weather is winter. It has been winter for a while and will probably remain so for a while yet. So quantifying it in precise degrees seems unnecessary. Right. Very good.
In there.
As I know you are all about plaque assays, I wanted to share with you this movie of a plaque being formed, attached. It shows a single well out of a 96 well plate filled with a monolayer of vero cells. To each of these wells, I have sorted a single HSV1 infected fibroblast and imaged it for 4 days at 2-hour intervals. This particular HSV1 strain harbors 2 fluorescent proteins, ICP4 in green, an immediate early protein, and VP26 in red, a late protein. The movie starts about 18 hours after sorting, when viral spread has just begun. I like it so much that I spend way too much time staring at it instead of actually doing experiments. All the best, Nir Draymond, postdoc in the lab of Savaste. And this is a wonderful movie showing the...
I'm sorry, what does the letter say? I've just been staring at this movie.
It's mesmerizing. It truly is.
Exactly. Very cool.
Right.
It's spreading.
Very neat.
Yeah, it's beautiful. I could see where, and he sent it as a GIF, GIF,
right?
Which is a self-contained
GIF, right?
Well, it depends who you talk to, GIF or GIF.
I have this argument ongoing with my daughter. She says,
"Well,
GIF.
People online have arguments, GIF or GIF."
Yes,
GIF
is a peanut butter.
It's true.
Again, it's a fake word.
It is.
Graphic interchange format.
And
anyway,
the G in graphic is a hard G, so I think it's GIF.
GIF, okay. So you can, these are little graphic files that are very short movies. And so
short animation,
animations, you could You could turn any short movie into a GIF. So it's that. And you can see it starts very early in the top right, and then boom. So that that's where the first infected cell landed. It released viruses, and then they spread throughout the entire culture, right?
Mm-hmm.
Yep.
This must be under liquid. There must be spread is explosive.
It is.
Yes.
You can see it, Dickson?
You bet.
All right.
And for listeners who are into this, and for Near, if you aren't already aware of it, I refer you back to episode 68,
Yes.
Ode to a Plaque.
Ode to a Plaque.
Where there is a similar movie from a vaccine. We discuss a paper that contains a similar movie of a vaccine.
Similarly mesmerizing.
Yes.
So since then, many people have done similar things. I know Paul B. Nash has done a movie with GFP VSV, because they sent me that. And then I saw on Twitter last week, Jason, the pox guy, Rich, Jason
Mercer.
He posted a movie of I guess a vaccinia virus plaque formation, fluorescent. And he said, "Oh, this is so cool! I love this." So I'm glad people are appreciating plaque assays. Alan, can you take the next one?
Sure. Darak writes, esteemed scholars of the podcast, TWiV has earned a place in academic discourse and in the history of how we communicate science. In TWiV 528, the annual recapitulation, Vincent said that all the podcasts are there forever. How? What happens if Primo gets bought out, Spotify, Vox consolidate all podcasts behind a paywall, Hurricane Sandy's big sister hits Manhattan, or other such disaster? What happens to the final cuts, the preliminary audio, the transcripts, the lengths, the letters? I'm sure you all have got a plan, but a friend told me about a workshop coming up titled It's the Year 2039, Where's Your Podcast? Provides a link. It's run by a group of archivists and librarians who are making Preserve This Podcast, a podcast series about preserving podcasts, coming out soon.
And what's going to happen to them?
Hopefully that'll be well preserved. The in-person workshop is on Friday, March 22nd in Hell's Kitchen, Manhattan.
We know.
They also have a zine on, I didn't know zines still existed, on preservethispodcast.org with recommended practices and a survey of podcaster practices. I hope there's a robust system in place, or that this resource can help y'all improve your current plan. Also, I think professional librarians would be excited to help archive the shows. Best of luck to Rock in California, where it has been unusually rainy.
It certainly has.
That is an interesting problem.
It's podcasts all the way down.
Yes.
I was thinking of going to this, but it's a Friday from 11 to 2, and I have a podcast at 2 p.m.
You have a podcast to do.
You're right, but I'm going to see if I can learn from their website something, because I don't have a plan yet in place for making these go forever. So I'm curious of theirs. But it's a good point. You know, some formats that we used to have years ago, you can't even read anymore, right?
Come on CDs.
Digital archiving in general is a complicated problem.
But so is paper if you burn everything, right?
Well,
everything is.
Okay,
but 2 billion years from now,
nothing,
come on.
Yeah, nothing lasts forever, but digital stuff goes bad a lot faster than a lot of the old stuff, because you need to take the play medium and the recording medium into account.
But in our case, we can't do it any other way, right? You want us to make tapes? That wouldn't be good.
No, that's not going to
make CDs.
Make CDs.
CDs have a limited
CDs have a limited lifespan, and who's going to have a CD player?
Yeah.
Someone gave me a CD the other day. I was at Charlie Rice's. He gave me a CD.
Wow.
What are you going to do with it?
Well, I happen to have a 10-year-old computer here, which has it, but nowhere else. My laptop doesn't have it at home. But so it's actually an interesting movie that I want to show everyone at some point.
Right.
But this is a good point. I really need to do something, but I can't. If someone wants to go to this, it would be great. If some listener in New York wants to go to this, you could tell us about it. But I can't go because I have to record TWiV at 2 p.m.
Yes.
And I don't want to miss it.
We're inflexible. We won't do it any other time.
Although we could do it at 3, I guess, right? I could get up here by 3 from Hell's Kitchen, right, Dickson?
Listen, I get out of work. I just teach down there, so I could just...
Is that Hell's Kitchen?
Walk to it. I could walk to it.
Hell's Kitchen is west.
In the 70s.
West of 8.
I teach in the 60s.
No, Hell's Kitchen is a little further down than that, isn't it?
Well,
yeah, I thought it was 50s.
Yeah.
Why don't we look it up? But in the meantime,
right,
the announcement for this thing Bring your hard drives, your laptops, and your Dropbox password. We're going to preserve your podcast. No, I don't want them to touch my podcast. You can tell me how to do it, but I'm not going to bring my Dropbox password. That's where they want me to archive it? That's not an archive.
No, Dropbox is not going to. No, that's not going to. Because what happens when they get bought?
Precisely. Okay, anyway.
Yeah, so Hell's Kitchen is west of Midtown, basically, around the Lincoln Tunnel up to Columbus Circle, west side of the river.
Okay.
Pretty broad range.
Brianne, did you do an email yet? I don't remember.
Have not.
Okay,
no.
Please take Peter's.
All right. Peter writes, and he is a retired physician. Owing to the increased interest in a viral cause for Alzheimer's disease, why is it difficult to develop a vaccine for the herpes simplex virus? I understand that many pharmaceutical companies have given up trying. Thanking you in anticipation. Kind regards, Peter.
Well, I looked this up. There is a type 2 herpes simplex vaccine in trial. And there's a lot of activity. If you look in Fields Virology, they say it's a high priority, and many companies are working on it. But the most advanced is HSV2, because this is the genital infection virus, and in many countries with HIV, high HIV, that's a big problem.
Yes, it is.
So that is a reason for that. But there are lots of type 1 candidates as well. The only issue I thought of is you'd have to give it quite early in life, because you typically get infected in your first year, right?
Right.
So that's possibly an issue. But there are companies working on it. I don't think they're giving up. I'm not sure that's...
I think there has been difficulty in even, regardless of when you might give it, there's been difficulty in just making a vaccine that's efficacious. And I don't know why. I don't know what the problem is.
Well, there's a lot of literature on it. You could certainly go look.
Yeah, I mean, we need to know what the correlates of immunity are and how to induce them and all
remember,
that.
Shingrix just came out.
Yes.
Shingles vaccine is a single glycoprotein. We did the paper, almost 100% protective in the clinical trial. It's remarkable. It's going to replace the other shingles, the infectious attenuated shingles vaccine. So I wonder if a similar approach...
So vaccinate against reactivation rather than vaccinating against initial exposure.
Yeah, that could be. Now, speaking of Alzheimer's, go ahead.
I was just going to say, if you want to get the Shingrix vaccine, get your name on a list at your pharmacy.
It's in
and
short
the priority
supply.
will be priority is given to people who already had the first dose. You have to have the second one 2 to 6 months later.
Wow. They're lining up for it.
Oh, yeah. Great.
Speaking of Alzheimer's, yesterday on TWiV, we did a paper, which is just amazing, showing correlation between P. gingivalis, an oral bacterium that causes gum disease, and Alzheimer's disease.
Wow.
It's amazing.
Yes, I've heard about that paper.
The bacteria is in the brain of almost 100% of Alzheimer's victims. When you floss or brush, you get a transient bacteremia.
Right.
And they think that gets into your brain. And then they showed in mice, you can give them this bacteria. It causes and by the way, the bacteria in the brain of AD patients is in the plaques, and you can give mice this bacteria and it causes brain plaques. And then they developed a drug that, the reason that this bacterium makes plaques is it has a protease that cleaves something and makes plaques. And then they developed an inhibitor of the protease, and it blocks plaque formation in mice. It's in clinical trial right now in people.
So this is 2 other kinds of plaques: dental plaque.
Yes. Yes.
Yeah.
And mental plaque.
Oh yeah, that's right. That's right.
We've just talked about viral plaques, dental plaques, and Alzheimer's plaques in the same.
Vincent has a lot of plaques on his wall.
But so there's going to be multiple AD etiologies,
right?
Possibly herpes simplex virus.
It is overdetermined. Yes.
Bacteria. But this one looked very good. All right. Who hasn't read a Dickson? Have you not read a letter?
I have not.
Trudy, please.
Trudy writes.
No, you didn't just arrive. You arrived some time ago.
Yes.
Right?
Dear TWivers, I wanted to write this email as a personal thank you and testimonial to Susana Lopez and her co-authors for writing this wonderful children's book called Paul Has Measles. I've been reading the book to my almost 5-year-old, and she really loves it. On a ride home tonight, she asked me out of the blue, Mom, when are the leukocytes coming? At first, I was very confused because the question came out of nowhere. So I asked her what she meant. She said, when are the leukocytes coming in my blood so they can get rid of germs? For context, my daughter has been suffering from an ear infection and her ear still hurts a little. So I will leave you with that as I pick my jaw up off the floor. I cannot recommend this book enough. Best regards, Trudy.
Nice.
Excellent.
Wow.
Excellent.
Wow.
Then we'll make a new generation of kids who grow up to be pro-vaxxers.
Yeah.
Excellent.
Yeah, I guess that's the intent, right?
Yeah.
Yeah.
And
sure.
I continually...
Well, and to understand immunology.
Yeah.
Well, she put 2 and 2 together and got 5.
Yeah.
I
That's great.
get emails from people in various countries who want to do a translation.
Nice.
I got last week someone who wants to do it in Swedish. And so Susanna didn't have that, so she got another one. So it's cool.
Lovely.
That is very cool. Can they make a sequel that talks about how antibiotics don't treat measles?
Exactly.
Or any other viral infection?
Right.
Now, we have done some papers where antibiotics actually impact virus infections for other reasons, right? Which really bothers me when we do them, because then people say, oh, look, I told you antibiotics could be used to treat virus infections. Okay, it is time for some picks of the week.
Uh-huh.
Let's start with Brianne. What do you have
All
for
right.
us?
I have an article that is in a PLOS blog that I found online. It's called Yes, I'm a Scientist on Twitter. Stop Judging Me. And it talks about some of the benefits that scientists can get from being on Twitter. I find Twitter to be incredibly beneficial, both for helping me find papers early and sort of connecting me with the field and making me not always feel as isolated from some of the other names in the field. So I find Twitter phenomenally useful for my work. And I thought that this really described some of the benefits quite nicely.
Cool.
I'm happy to say I was on Twitter a long time ago, and everyone used to say, what are you doing that for?
Back before it was cool.
What are you doing that for? I'm very glad that a lot of scientists are on it now. By the way, this week I was called an emeritus virology professor on Twitter.
Oh,
Someone is retiring me.
Oh, dear.
Oh, okay.
Yeah, well, it's okay. I don't mind. It's because I know I'm not, right, Dickson?
Exactly.
Alan, what do you have?
I have a set of graphics from the Wall Street Journal. This came out a couple of weeks ago, and it's graphics showing the impact of vaccines. And as Kathy has pointed out in the show notes, some of these I'm not quite sure why the data line up the way they do or why they've chosen particular time points as having been the origins of these vaccines. But the first one is really dramatic. It's measles. And so what they've done is they've taken CDC data from all 50 states and lined that up and then plotted over time incidence of the indicated disease, and then they show when the vaccine is introduced. And so you see these squares of different colors indicating the incidence of measles in all 50 states. And then the vaccine is introduced, and within a couple of years, it just goes to all light blue and white, which indicates 0 or very few cases. And then you follow down the page and you see some of them, like hepatitis A was under some control, but then the vaccine introduced and it just fades away, and mumps fading away. Pertussis is a little weird because there's a gap in the CDC data. They weren't reporting those data for like 20 years. So there's just this blank section, and then pertussis is gone. And I'm also not sure exactly when they mean that the vaccine was introduced.
Because they say 1914, but Wikipedia says there was a vaccine developed in the 30s by a woman last name Denmark. Yeah.
Right. So I'm not sure. Some of these I'm not quite sure what the underlying assumptions were, but the graphs are overall valid. Polio is another pretty dramatic one. And I think
polio even shows the epidemic.
Yes.
Mm-hmm.
Okay. It's sort of it shows it simmering in the 30s
worse.
and 40s.
Right, getting worse in the 40s and 50s, probably as a result of sewage systems coming online. And then vaccine introduced, and it's just blam. And there it goes.
That's also...
smallpox has an interesting trend, too, because the vaccine was around essentially way before the graph. And you don't really see the cases going down until about 1940, 42. And then they just drop off precipitously. This graph is on a different scale for some reason. But I think that's
I
because...
looked that up to see if I could find a reason for that, and it wasn't obvious to me. You found a reason?
No, I hypothesized one. I had a similar experience. I was like, did we have some big smallpox vaccination campaign? We didn't, but we did have a big military mobilization. And I think that may have, and there was a big push for public health after World War II as well. So I think it may have just been a ramping up of production of the vaccine for those things, is my guess.
This polio late 40s, 50s is baby boomers. A lot of new kids born, and that's that big tick also, besides sanitation. It's a cool. So unfortunately, everyone listening is pro-vaccine, but
already understands this. But
how do you get this to more people to say, look, if you don't want to vaccinate,
well, you publish it in the Wall Street Journal.
Yeah, right.
Yeah,
but
which is read by people in Congress and other folks who may not quite get it.
It's not read by enough people who are anti-vaccine. There have to be other ways, like a children's book, I guess.
Yeah.
Rich, what do you have?
So I just read a book that was recommended to me and, in fact, even sent to me by my buddy Ed Niles called Gene Machine, The Race to Decipher the Secrets of the Ribosome by Venki Ramakrishnan. And he was one of the three people, along with Tom Stites and Ada Yonash, who won the Nobel Prize for figuring out the atomic structure of the ribosome. And so this book is it's his own memoir and sort of science biography. So it tracks his career through science and also gives a good description of what the ribosome is and what the ribosome does and how all of the individuals involved and other individuals studying the ribosome contributed ultimately to the description of the atomic structure of the ribosome. And it's a really easy, quick read. He also has comments that I really appreciate on the role, for example, of competition in science and what it was, what the interactions amongst the different people were like. So it's a good read.
Cool.
Got good reviews here. By wow, his telling is laced with wisdom spun from a remarkable life story. He takes up the baton from Double Helix, and his tone is disarmingly candid, sometimes disquietingly so. I'll have to read this. I will have to read.
It's very good. And, there are a lot of people who've spent their lives working on the ribosome. And the Nobel Prize can only go to 3 living individuals. And so there was some controversy when the prize came out as there always is, really, about, who deserved it or whatever. And he's very good at describing what went into that decision. And it's clear that it went into that the prize was given for solving the atomic structure. Very specifically, and for the individuals who really pioneered the techniques that were necessary to solve such a large structure, because it's probably the biggest structure that's ever been solved. So he's very, very clear about that, and that was really good. I actually wrote him quickly afterwards because my scientific life has overlapped with many of the characters involved in the book. And so I thought I sent him some pictures, that one that you've seen was from 76 with standing with Harry Nuller outside the pub and a couple of others. The one you took of me with Joan at the ASV meeting, and just told him I enjoyed the book and thought he'd appreciate the pictures. He was great. He wrote me right back.
Cool.
Thanked me for the pictures and stuff. So I really recommend this. It's a very, very candid, very readable description.
All right. I'm going to get it.
Sounds really cool.
Dickson, you should write a book about your career.
I don't know. Stop.
You could write an exciting book.
Yeah, well, I hope I am writing an exciting book, but it's not about my career.
Okay. Well, what do you have for a pick then?
Well, I have a pick, and I noticed that there's a line cross through the second pick, but we'll come back to that. I thought I had one pick first, and I ran across these pictures of leaf hoppers, and they are just unbelievable pictures of weird. I mean, you couldn't make these up, basically. They look like some machination of a Salvador Dali on steroids or something. They're highly selected for whatever those structures are that bud off of them. And to live their life just by looking at these pictures, can you imagine what the selective pressures were that led to these shapes? And the answer is no. I could not. So that's independent of my second pick, but I think I should reserve that for next time because it relates to what these things actually do. And one of the things they do is transfer viruses from plant to plant.
Oh.
So here's the second art, and I ran across it totally by luck because I was looking for something completely different. And the cover of PLOS Pathogens this month has a picture of it's not a plaque, it's an infected region of a plant from viruses that were transmitted to it by leafhoppers.
Okay. So you're going to save that for next time?
Yeah.
All right.
Maybe we'll review the article.
The leafhoppers are pretty cool.
There are, though.
They are. These things are really wild.
They're like little kids' drawings of, I think I know what an insect looks like, and then they draw these, phantasmagorical stuff. These are real things.
Kathy, what do you have?
Oops. I just clicked away because, anyway, I picked World Watershed Map. So in the keeping of our map arc, this shows a whole series of watersheds from around the world. And the thickness of the line has to do with how wide or deep or both the streams or rivers are. So yeah, width to indicate the hierarchy of streams, and then they're grouped by color. So it starts out with a map of the U.S. and then a map of the world and Africa, and they're just really beautiful.
These are gorgeous.
Yeah,
I agree.
Check them out, and look at Great Britain and Ireland, and how many rivers there are for that area compared to say, Germany.
That's right.
It's really fascinating.
Yeah.
Websites are getting just full of ads.
You're right there.
Yeah.
I mean, and it's worse and worse.
You are right.
Even the Times, I subscribe to the Times. I still get all these ridiculous things. You read a paragraph and then there's a bunch of ads and you read a paragraph.
It's part of the death spiral because the ads are paying less and less, so they have to have more and more of them, and then they're less and less valuable, and this is just continuing, and it's going to end.
It's horrible. It's horrible. Really ruins your reading.
It is not a sustainable method of supporting the web, and everybody involved knows that, but they don't have a better idea, and so they're stuck just amping up the number and the intrusiveness of the ads.
And they say, you have to see this blah, 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 blah. No.
Right.
I'm doing something else right now. This is the problem. All right. So my pick is two picks. They're both related. The House Committee on Energy and Commerce this week had a hearing on the confronting a growing public health threat, measles outbreak in the U.S. This is great. Okay, and they had two witnesses, Nancy Messonnier, who is director of the National Center for Immunization and Respiratory Diseases, and Anthony Fauci, director of NIAID. And this is great that they had a hearing on this. It's something I never thought would happen. And then there's there is a letter from the American Society for Microbiology. ASM applauds subcommittee hearing on measles outbreaks in the U.S., and they describe it, summarize it, and talk about the history of measles and vaccination and surveys that have been conducted. So this is great that they did this and make it more visible. Okay, so the more you can do, the better. And I don't know, you could actually watch the meeting because there's a video player there and see what happened. But I am sure that the two witnesses spoke strongly in favor of stopping this and increasing immunization rates, which is what should happen.
I mean, people have an answer for the requirement that your children can't go to school unless they're vaccinated. That's developed into a large homeschool industry, which I think has really undermined the intellectual capacity of this country in some respects, because people are avoiding other opinions and they're not getting involved in debates, and they're
part
isolating.
of the whole bubble syndrome.
Yeah, they're siloing themselves into these small knit groups that all think the same way, and I think that's what you see when there's an election, for instance, when it comes out. There's such big differences between groups. You see all these things coming out at the wrong times, and I'm really against that entirely.
I had an experience in my class. We do a little virus in the news at the beginning of each class, and so I did one right at the beginning of the semester about the measles outbreak, and I said, and you can get a measles vaccine at the University Health Service, particularly if you were homeschooled, you might not have gotten this vaccine. And someone pulled out a piece of paper and, made a note at the time. And I thought, well, okay, maybe we'll get one person vaccinated from that. So.
Or you'll get a nasty note sometime when you least expect it.
I think you guys, was it you guys talking on TWiV? I can remember where I was when I heard it, where I was walking about young children or teens, maybe 15 or so, wanting to get vaccinated, but they can't because their parents, because they need parental consent and their parents are anti-vax. Maybe it wasn't on TWiV. Maybe it was.
I've heard that story.
Yeah. So that's an issue. And so I didn't say it to my students in my class, but I assume they're all over 18. And so that wouldn't be an issue for them if they want to get it.
Dear.
So we have a listener's pick, which is related. It's from Justin, who sends a Bloomberg article. Facebook says it may remove anti-vaccine recommendations. The only problem with this is the may. It should. Just do it. Because on Facebook, information discouraging people from getting vaccines goes viral. And it's thought that this has contributed to an increase in measles outbreaks, right? There's no reason for that to be there at all. Now, Zuckerberg says he's not doing anything wrong. The world needs Facebook, and he doesn't have to fix anything. And this is part of the problem, right? Unbelievable. This is a company that should be broken up. It should not exist. It's too big, and they can't police what's going on. My pick for the next TWiV will be an article about the people who do the policing on Facebook. It's so screwed up. It's unbelievable. They have 15,000 people who are looking at posts and saying, yeah, this is okay or this is not okay. It's unbelievable. But anything anti-vax should be off Facebook. And every time I post something which has vaccine in it, you get thousands of people, maybe not thousands, many people saying you shouldn't get this vaccine, you shouldn't get that vaccine. It's ridiculous. It's ridiculous. Now, I understand that it's freedom of speech, but when you have companies that say we have something better, for example, so they have to do something about it. So this article says they may do it.
Just to clarify something about freedom of speech.
Oh, yes.
This is a freedom granted from government censorship.
Right.
If you are posting something on a private company's website, that private company can make whatever damn rules they want about what you can and cannot post. And if they say otherwise, they're wimps.
That's right. You're right. We've mentioned this before. That's right. Freedom of speech is just for the government censorship. Yeah. That's right. So they should shut it off. The guy's a wimp. He just wants to make more and more money for his company. And everything they do is damaging to this country and the world. And I am thinking really hard about being on there. I mean, I think it's important to spread our messages there, but I hate doing it. And I don't
quit there years ago.
I don't spend time there. I post what happens. And man, it's so toxic. It's unbelievable. Thank you, Justin. All right. That is TWiV 537. If you listen on a player, please subscribe so we know how many people are listening. And if you want to send questions and comments, they go to twiv@microbe.tv. And if you'd like to support us financially, we'd love your support. Microbe.tv/contribute. There are a few ways you can do that. You could give a fixed amount of money, $1 a month, either at Patreon or PayPal. We'd appreciate that a lot. Dickson Despommier is at parasiteswithoutborders.com and thelivingriver.org. Thank you, Dickson.
I'm always glad to participate.
Did you have a nice class?
I did. We're moving along. We're creating a new city called Fordomopolis 2.0.
I bet you're teaching at Fordham.
I am.
Because at Columbia, that might not be a great course.
No, they wouldn't allow it here, I'm sure.
Why don't you teach a similar course at Columbia?
They haven't asked me.
Well, ask them. See, I didn't ask them to teach. No, I did.
Yes,
I asked
you did.
them.
Yes,
They didn't
you
ask
did.
me. I asked them, could I teach this course?
No, I did. I asked to teach down there because nobody wanted me to teach up here anymore.
Well, how do you know if you don't ask them?
No, I did ask. I asked.
It'd be closer.
I asked
anyway.
the director of
Oh, I
curriculum development whether I could stay on after I retired, and he didn't give me an
Well,
answer
you should ask at
until
Morningside.
after I
Tell him
retired.
you should be nice to Dickson.
You know, I forgot to mention that actually.
Well, anyway, they have a website where you put this, right?
Yeah, you helped with it. And,
Fordomopolis.com.
yep, and you're going to have to help him again because
Cool.
yeah.
Brianne Barker is over at Drew University on Twitter. She is BioProf Barker. Thank you, Brianne.
Thank you. It was great to be here. My students are going to be really excited. They loved this paper, and they are going to be happy to hear more.
They should listen, right?
They should.
I had a letter from Brianne on TWiP. Was it TWiP? No, TWiM yesterday from Brianne because we gave away a book.
Excellent.
And I saw it and I said, oh, Brianne is entering the... Oh, but it's not that Brianne.
No, it wasn't me.
Kathy Spindler is at the University of Michigan in Ann Arbor. Thank you, Kathy.
Thanks. This is a lot of fun.
Rich Condit's an emeritus professor, University of Florida, Gainesville, currently residing in Austin, Texas. Thank you, Rich.
Sure enough, always a good time.
Alan Dove is at turbidplaque.com. You can find him on Twitter as Alan Dove. Thank you, Alan.
Thank you. It's always a pleasure.
I'm Vincent Racaniello. You can find me at virology.ws. Thanks to ASV and ASM for their support of TWiV, and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral. Some of these are references I don't understand. Like, I know Tangled Up in Flu, that's Bob Dylan, right?
Right.
And quick ribosome to the bat cell. Is that from Batman?
Quick ribosome to the
Batman.
bat cell.
Yes.
Instead of Robin, quick Robin to the bat cave.
Yes. Yes.
No.
No, that's the.
No. Bat flu.
Oh, that's
flu.
Batman.
Yeah.
Yeah, we need more non.
Bat flu.
Yes. I had the paper on my desk today, and I think that every person who walked into my office sang that at some point.
No. How many nanas do you need?
No. 8, right?
Whoops. It's not all there.
No.
Hey.
That'll do. Yeah.
No. Bat flu. That's probably okay the way it is. I like mine, but it's about gulls, not.
Yes.
It's true.
We'll use it another time. Breaching borders. Didn't we use Bat Out of Hell?
I think we must have.
I
Right?
don't remember when, so I just put it there for the heck of it. Or for the hell of it, actually.
Wait, these have to be Anna,
Yeah, they have to be nanas, right?
Ah,
Exactly.
yes.
Anna.
And then you need 4 more, right?
Yep.
That's a long title.
Yeah, but I
No, you
think
could
you
just
could
do
get
4.
away with just the 4. That's
Just the 4.
Na, bad flu. That'll do.
Yeah, that's good.
Yeah.
Instead of 8.
It'd have to be because otherwise it would be too long. Yeah, so they did this in Batman? Who did
No,
that?
it's the music.
Yeah.
It's the
The
background
music,
music.
the music.
the theme, and
Oh, so someone is actually doing na.
No.
No?
No.
no,
That's
the
how
music sounds like that.
Dun, 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 bat flu.
Except it's Nana.
It's Batman. And
Yeah.
Nana, by the way, means small.
Is anybody going to know what this is?
Yeah.
This is cultural.
If you play
Stuff.
this part of it.
By the way,
Right.
breaching borders and boundary issues could apply to both papers.
Yeah, that's their
true.
plus.
Yeah, let's pick one of those. If everyone likes them. Breaching borders or boundary issues.
Of those, if we're going to pick one of those, I'd go for boundary issues because that just has a little more emotion in it.
Yes,
You know.
and less political.
Yes.
True.